Thank you for inviting me to give this talk. So I decided to focus today on sort of talking a little bit about some of the genomic data that's coming out of sequencing pancreatic cancer. This is actually, I don't know how this works. Is there a pointer? Ah, there's a pointer. Here we go. This is actually the new building will be starting January 2015 at MD Anderson. The 7th floor of this will house the Pancreas Cancer Research Center, which I'm the director of, and then we'll have the Institute for Personalized Cancer Therapy, the molecular diagnostic labs, the moonshot programs across various floors. So it's really a great composite building to be in, surrounded by people, all of whom are working feverishly against cancer. So very quickly, this is some data that the Pancreatic Cancer Action Network published earlier this year in a report. I'm the chair of the Scientific Advisory Board, and it's really astounding in terms of the numbers of cases that are being diagnosed with pancreas cancer each year. Because of early detection, working in other diseases, and unfortunately not so much in pancreas, you can see there is a dramatic rise in the number of cases, such that by 2030, it's predicted to be the second most common cause of cancer-related deaths, and if smoking keeps going down, which we hope, and lung cancer rates go down, then pancreas actually might emerge as number one someday, which is really kind of a fearsome prediction. Unfortunately, we haven't really made a dent in terms of the impact on this disease, and obviously you're familiar with the gemcitabine phase 3 trial data, 1997. This is FOLFIRINOX, and I haven't added the gemabraxane impact trial data that came out just recently in New England Journal of Medicine. And the data are somewhat similar to the FOLFIRINOX, probably actually a little bit less, but overall, if you just look at the survival impact we've had, in 14 years since the gemcitabine trial, we've improved the median survival by 5.5 months, so really haven't had much of an impact. We now know a lot about the genomic landscape of pancreas cancer. This was from a 2008 paper published from the Hopkins group, Vogelstein, Kinsler, Velculescu et al. And you can immediately see when you look at the landscape that there are these big four genes that stand out as being recurrently mutated. This is actually sort of a word cloud from Andrew Bianchian, who led the ICGC effort, the International Cancer Genome Consortium effort from Australia, in sequencing about 100 plus pancreas cancer. His paper came out in Nature last year, along with folks from Canada, from Baylor, and other countries. Hopkins also contributed cases at that time. And you can see, when you look at this, that the big four just literally stand out. It's right here. This is KRAS. This is really the biggest player in pancreas cancer. You have P16, SMAD4, and P53. But then all around you have all this incredible molecular heterogeneity, these genomic molehills, as we call them. You have these 4 mountains, and then you have this huge smattering of molehills that are everywhere. And I guess the question for us as we try to implement many of these molecular targeted therapies is there something we can do going forward? Once again, KRAS is obviously the big enchilada in pancreas. This is the ICGC effort that combines, that looks at many different cancer types. So pancreas cancer was contributed from Canada, US, Australia, Italy, several different countries contributed samples for this. And here's KRAS right here. And at least there is now a national effort at trying to do something about RAS mutations. In the past, drugs that have targeted RAS or have targeted enzymes that processed RAS post-translationally haven't really done very well in the clinic, unfortunately. And now there is this concerted effort led by Frank McCormick. Some of you may have received this email just this past week. This is a new project called Solve RAS project that's going to be led by Frank. It's going to be housed at the Frederick National Lab under NCI oversight, and Dr. Varmus, the NCI director, has committed $10 million to this program. There are many investigators around the country who are involved in this, like Channing Dar, et cetera, but Frank will be the main person leading it. And the idea here is can they find RAS therapeutics using new unconventional methods, and obviously the impact of this, while it'll be felt in other RAS mutant cancer types, will probably be most profound in pancreatic cancer, where this is such a common mutation. So at least something is being done about RAS, the big enchilada in pancreas. But when you look at the molecular taxonomy of pancreas cancer beyond KRAS, and again, a lot of the slides that I'm going to show today have been kindly given to me by colleagues for this purpose of this talk. So Andrew Biankin lent me this particular slide. You can see again this molecular heterogeneity being reflected. The width of these bars don't necessarily correspond to the frequency of mutations. This was just sort of a cartoon he made up. But the idea is again to now pick out these individual molehills and say, can we try to target them somehow with specific therapies? So, and some of these are already either in clinical trials, or there is data from other cancer types suggesting that there's evidence that this might work. For example, there is a proportion of cases, about 6 to 8%, that are KRAS wild type. And so one could envision, just like you do in colon, could you put these patients on an erlotinib-based therapy? There is very new data now coming out that there's a small proportion of cases, about 2% of pancreas cancers, and I'll talk about this later, that is HER2 amplified. Could these patients be treated just like you would treat HER2 amplified patients? And of course, we have the 5 to 7% of patients that have DNA damage response gene mutations, like in BRCA2, RPA1, PALB2, etc. And these patients, we know from anecdotal data, respond dramatically to DNA damage, to DNA damaging agents, especially platinum-based therapies or PARP inhibitors. And the idea is, can we now actually make this the standard of care? And then others for which there is preclinical data, such as MET amplification. There's some work from Elisa Gianoveri's lab in Netherlands showing MET amplified pancreas cancer lines respond well to crizotinib and may actually sensitize cells to gemcitabine, and then PTEN loss and mTOR inhibitors. Some of this is in mouse models, not yet in the clinic. But definitely there are points here, vulnerability nodes here, that you can start thinking about in the context of pancreas cancer beyond KRAS. So in the ICGC cohort of cancers that were treated, one of the things is there's a lot of cases that have clinical data associated with them, and so you can actually go back and ask, how did the patient respond to the therapy, and correlate that with the underlying genetic data, which is really nice. So it turns out that in their subset, they had follow-up data and treatment data for a subset of 27 or so patients. 10 of these patients received, at that time, interestingly enough, even not knowing the genotype, what they call on-phenotype treatment. So this was treatment that was given that you would think about giving if you actually knew what the genetics of these cases were. And then 17 unselected patients who were given just, what was at that time considered standard of care. And if you look at the Kaplan-Meier survival, it's very interesting. The on-phenotype patients had a median survival of 21 months, including 6 that would be considered exceptional responders, which meant they were alive 3 years after diagnosis, 3 or more years after diagnosis of recurrent disease. That's really remarkable. And then, in contrast to that, these unselected patients, you can see, had the usual response to pancreatic cancer. So one of the big, not big in terms of the numbers per se, but the obvious sort of responses that came up was this pan-genomic instability phenotype, or genotype I should say, where you could get discernible response to platinum-based agents. So and these are the so-called circus plots that denote mutations and copy number alterations. And the first thing you see when you look at the circus plot is that this particular patient, this is an actual individual tumor, you can see that there is this dramatic increase in the number of translocations and losses. So this is a pattern that we call pan-genomic instability. And this was this patient who had surgery, got adjuvant gemcitabine, had a recurrence, had second surgery, and then was given FOLFOX, a platinum-based regimen, and had a real dramatic drop in their CA19-9 and responded really well. So in their series, they had 4 patients who got this sort of on-phenotype response, and all 4 of them are still alive, and only one of the 5 that was given with this particular genotype was not given the platinum-based therapy. Only one of those 5 is alive. So really sort of striking anecdotal data here suggesting that this pattern of pan-genomic instability can be used as a guide to therapy using platinum-based regimens. We also have published a similar story. This was a gentleman who actually developed metastatic cancer here in the supraclavicular lymph node in the lungs, was given a mitomycin C-based therapy, responded really well, and we found out later on, this is published, so I'm really skipping through this quickly, that this person had a biallelic mutation in PALB2, which is a gene which is called partner and localizer of BRCA2. It works basically localizing BRCA1 and BRCA2 at sites of double-strand breaks, and so when you have mutations in PALB2, it's essentially a phenocopy of BRCA mutation. And this particular individual stayed alive for 5 years after progressing on first-line gemcitabine, was very grateful that we were able to keep him alive for this amount of time. It's a really striking example here again, where you can use the underlying genotype to have these sort of extreme responses in pancreas cancer. So once again, even without necessarily knowing the underlying DNA damage response gene mutation, you can use this sort of pan-genomic analysis to predict which individuals might respond to platinum-based therapies. In general, in pancreas cancers, you do have some structural variations, but it's not dramatic. But when you start looking at this subset of patients that have this high prevalence of structural variations, about 8%, which matches to the 5 to 7% BRCA2, and then you add in a few percents of PALB2 and RPA1 and ATMs. So this is about the right percentage. So about 8% will have this pan-genomic instability, and when you actually start looking at individual cases, here's someone who has germline BRCA mutation. This is someone who has somatic BRCA mutation, and again, I point out that when you have somatic mutations in conjunction with germline, so you have biallelic mutations, the intensity of the structural variation is really dramatic. Here's someone with a somatic RPA1 mutation, again, just dramatic pan-genomic instability. Here's a germline ATM and a somatic ATM. And then going back to the actual responses, many of these patients here had pretty good response to DNA damaging agents in this ICGC cohort. This is new data, again, coming from ICGC cohorts about when they go back and look at their patients, about 2% have HER2 amplification by FISH. Again, because this is tied to clinical data and presentation, these patients, these HER2 amplified pancreatic cancers have a somewhat unusual presentation. They have lung and brain metastasis. Uncommonly, they have liver metastases, so the metastatic spread pattern is a little bit more different, and in fact mimics HER2 amplified breast cancer, which is kind of interesting. So will these patients have a response to Herceptin? We don't know. There's only one patient in the ICGC cohort that got Herceptin and survived for, I think, over a year on Herceptin therapy, but I think this is a trial that needs to be done. So the question that you might ask is, well, how would we ever accomplish a trial with, say, with Herceptin in pancreas cancer that has only 2% frequency of amplification? So now this brings me to another concept that I want to put out there. This is actually a concept propagated by Dave Solit at Sloan Kettering. Again, Dave has very kindly donated some of his slides to show at this meeting. So he's talking about this concept called basket trials. And so what are these basket trials? This basically came from a trial that was going on at Sloan Kettering, which was basically failing, and in fact failed, except for the fact that they had this 1 or 2 patients who had this extreme response. So this trial was basically a phase 2 trial of everolimus, and they only had 2 patients, one of whom was a patient with a bladder cancer, metastatic bladder cancer, who had a dramatic response to everolimus. Metastatic sites here completely melted away on everolimus therapy. And so while the sum total of the trial failed, Dave was very interested as to why this particular patient responded. And so once again, they took the tumor, did whole genome sequencing, copy number analysis. Turns out this patient actually had a mutation in TSC1. You can see here, this is a normal tissue. This is a paraffin sample, frozen tumor. There's a point mutation in TSC1, and there's a copy number alteration on the other allele. And basically, so there was complete loss of TSC1 function. And this person, obviously, as a result of that, had aberrant activation of the TOR signaling pathway, was ripe for everolimus therapy. So based on this, they're now running a trial where study entry is restricted to patients with TSC1 and or NF2 mutations, because the other thing that came out of this trial was that she also turned out to have an NF2 mutation. In fact, the 2 patients who responded had both NF2 and TSC1 mutation. So when you look at a given cancer, be say pancreatic, bladder, lung cancer, whatever, there's 2 ways of doing these sort of trials. One is you take, the inclusion criteria would be the cancer by pathology, so lung cancer, non-small cell, or pancreatic adenocarcinoma, and you genotype these patients and you allocate them to particular drugs based on profiling results. So you could have EGFR, BRAF, MEK, AKT, TORC1, whatever. But there are a few problems with this approach. The first is that when you start these trials, the drugs are often not necessarily the best one that's available. And at MD Anderson, there was a battle trial that was done in lung cancer. And when this trial started, sorafenib was used as a quote-unquote RAF inhibitor, but then subsequently much better RAF inhibitors came along. This was not really the best one. Many of these trials require a process called adaptive randomization. And then during the course of this trial, if you're finding out that that there is a particular genotype that is associated with very good responses, it becomes unethical then to randomize patients who do have that mutation. And then the total number, this is probably the big problem for some of these little molehills you're talking about. The total number of patients is low. And this design, when you are including by tumor type, may not identify sufficient patients with these rare mutations, such as BRAF in lung or ERB2 mutations in ovarian. So here's an alternative approach that Dave and MD Anderson also is doing similar trials, the so-called basket study, where you basically don't include cases by the tumor type, but rather by the genotype of the tumor, the driver genotype of the tumor. And so you could, for example, have a BRAF trial, 3% of colon, 1% lung, 1% breast. Erdheim-Chester disease is a histiocytic disease, 50% of which is a very rare disease, but when you do have someone, there's a 50% chance that person will have a BRAF mutation. And then you basically divide them into different baskets, and they all get this BRAF inhibitor. And they've started these sort of trials at Sloan, and this is ongoing through the IPCT at Anderson as well. So this is a patient on a BRAF basket trial, lung cancer patient, a 1%er. This is a baseline, 6 weeks on a BRAF inhibitor, dramatic response. So if you look at the molecular heterogeneity of pancreatic cancer, it almost becomes a given that till Frank McCormick comes up with a RAS inhibitor or whatever, we are almost going to have to start thinking about this sort of basket approach for pancreatic cancer. And going back now to some of our Anderson data, this is, again, kindly provided by Kenna Shah, who's the director of the IPCT and Anderson. If you look at HER2 new amplifications, again, TCGA, ICGC, they all find this 2% HER2 new amplified cases. It would be incredibly hard to do a pancreas cancer specific trial with 2% of cases having this amplification and then enrolling enough patients to get some data. But once you start doing this as a HER2 amplified basket trial, now all of a sudden you start having a lot more cases to work with. Similarly, there are not too many HER2 mutations that have been reported in pancreas, but when you start looking at other cancer types that have rare HER2 mutations, you can start pooling these together in a basket trial, and then you can quickly get to a phase 2 type numbers. So I actually asked Kenna to do this, and she was very kind enough to look through the IPCT data and come up with these numbers. So, and it hurts me to say this as a pathologist, but if you were to do a pathology independent basket trial at MD Anderson, we already have 50 patients, and this doesn't even include pancreatic cancer. You already have 50 patients with BRCA mutations, 70 plus patients that have mutations in PI3 kinase, AKT signaling that would be expected to activate with TOR, 10 patients with MEK, 17 patients with TSC1. So very quickly, you can start adding these numbers up. And again, the idea here is this is a genotype-driven trial. You're not necessarily worried here about what the underlying pathology is. And then we look for responses. But here's a word of caution. There's always a caveat. Just because it walks like a duck, you have to be careful, especially with some of these missense mutations. Does it really functionally matter? And this is actually data from a paper in melanomas, in KIT mutant amplified melanomas, from Sloan Kettering, I believe, and basically there are certain mutations that are resistant to the drug of choice that you're using in your trial. So in this case, imatinib. For example, this D820Y mutation in KIT and the V654A mutation in KIT, these are typically secondary mutations that are resistant to imatinib, but when they're found even in primary tumors, these are patients you don't want to enroll in an imatinib trial because it's not going to work, okay? So and here you can see here the response. Basically, there's no response whatsoever, as opposed to, for example, the 576P mutation, which is a very strong responder to imatinib. So you have to be careful. Just because you're enrolling someone in a basket trial, just because they have a mutation in the drug of choice that you're working with, it doesn't necessarily mean that it will work. You might have to do some sort of functional genomics assay to make sure that your mutation, given mutation, is actually working. And we're doing this, at Anderson. It doesn't have to be that even that particular cancer cell type. If you can engineer the mutation in an ORF and just test it in some system like a BAF3 system or something, that might be enough. So one of the big challenges that we are facing right now is, so I talked a little bit about oncogenes, but what about tumor suppressors? We have this big subset of genes, is chromatin remodeling gene mutations that are pretty much everywhere in pancreas. This is the ICGC data. Similar numbers have already also been found by the SU2C sequencing data from Victor Velculescu. MLL3, ARID1, ARID2, SMARCA4, they're everywhere. And I'm not going to talk too much about this. My Sonal Gupta from my group actually had a poster, and I think he was one of the preferred speakers yesterday, so you may have heard about the MLL3 data. So basically MLL3 and as well as the other MLLs act as tumor suppressors in pancreas cancers. We have about 10% of cases that are mutated. We have a mouse model. So this would be potentially a great subset if you could have a targeted therapy. This was a paper we published in PNAS earlier last year with John Pollack at Stanford, looking at SWI/SNF alterations in pancreatic cancer at the copy number level. So even if they're not mutations, when you start looking at these at the level of homozygous deletions, which would basically be a complete loss of function, a surprising large number of cases have loss of function alterations. So this is a case, a xenograft that has a PBRM1 mutation, loss of homozygous deletion at this transcription start site, and when you look at the RNA level, there's a dramatic reduction in the PBRM1 levels. If you start putting these copy number losses together, about a third have alterations at these SWI/SNF loci. So that's an appreciable number. And then when you look at the expression signatures of knocking down, say, ARID1 function in a cell line model, the gene signature that you see is basically a mirror image of what you see with knockdown of the PRC2 complex, specifically the EZH2 genes. So EZH2 is activated in many tumor types, and so the idea here is could loss of components of the SWI/SNF complex be sort of correlating with aberrant EZH2 activation? This is a clinical trial that's waiting to happen. But before that, obviously, we need to do it in a preclinical setting or a co-clinical setting using xenografts and genetically engineered mouse models. There's certainly drugs available against EZH2 activation, but these chromatin remodeling genes, MLL3, which is a histone H3K4 methyltransferase, these are agents, or I should say these are targets that are ripe for testing as molehills in pancreas cancer, and I really encourage everyone to look into these as potential sort of targets that we need to think about. So very special thanks to Agi for inviting me. Thank you so much. I do want to say one thing quickly, which is that, as you know, November is Pancreatic Cancer Awareness Month, and so today is 2nd November, so think about how you can contribute to the field. But one thing that Agi, and she is, I think, the one who's really propagated this, is that it's not just a month, and that is, you have to think about pancreas cancer throughout the whole year and how you can impact this disease. So thank you, Agi. Thank you very much.